Welcome back, everyone. I remain Professor Dixon Achemugumusa. In the last module, we carefully examined carbohydrates, chemistry, structure, and biological functions. We learned how carbohydrates are classified into monosaccharides, oligosaccharides, and polysaccharides, and we explored their stereochemistry, ring formation, and conformational behavior. We also linked carbohydrate structure to function by studying important molecules such as starch, glycogen, and cellulose. That discussion helped us appreciate how small chemical differences can lead to major biological consequences. This idea will remain central as we move forward. You can find a link to Module 2 in the description below. Today, we begin Module 3, where our focus shifts to lipid. Lipids are chemically diverse molecules that play critical roles in energy storage, membrane structure, and regulation of biological processes. As we proceed, keep asking yourself how chemical structure determines biological functions. This will guide your understanding throughout this module. In this module, we will examine lipids from a chemical and biological perspective. By the end of this module, you should be able to 1. Explain the chemical basis of lipid diversity and classification, recognizing why lipids do not fit into a single structural pattern. 2. Describe the 8 major lipid categories defined by the Lipid Maps Consortium, which represents the modern framework for lipid classification. 3. Relate chemical structure to physical properties and biological functions, especially in biological systems. 4. Discuss the roles of lipids in membranes, energy storage, signaling, and regulation. 5. Apply lipid classification concepts to biochemical, physiological, and biomedical situations. These objectives will guide our discussion and help you connect lipid chemistry to real biological functions. Lipids are a heterogeneous group of organic compounds defined mainly by their solubility properties. They are insoluble or sparingly soluble in water, but soluble in non-polar organic solvents such as ether, chloroform, benzene, and hexane. This behavior reflects their hydrophobic or amphipathic nature, which is central to how lipids function in living cells. Chemically, lipids are composed mainly of carbon, hydrogen, and oxygen, although some classes also contain phosphorus, nitrogen, sulfur, or carbohydrate components. Unlike other biomolecules, lipids do not share a common backbone. They may exist as esters, ethers, amides, or hydrocarbons, which explains their wide structural diversity. Physically, lipids may be entirely hydrophobic, such as triacyl glycerols, or amphipathic, such as membrane lipids, that contains both hydrophobic and hydrophilic regions. This property allows lipids to organize themselves in aqueous environments. The physical state of lipids depends largely on fatty acid chain length and degree of saturation. In general, saturated lipids have higher melting points, while unsaturated lipids melt at lower temperatures. Chemically, lipids undergo reactions such as esterification, hydrolysis, saponification, oxidation, and hydrogenation. These reactions are important in digestion, metabolism, food science, and laboratory studies. Biologically, lipids are indispensable. They serve as a major energy store, structural components of biological membranes, signaling and regulatory molecules, hormone precursors, and agents of protection and insulation. Understanding these roles begins with understanding their chemistry, which we will now explore step by step. To properly understand lipids, we must first address how they are classified. Lipids display extraordinary structural diversity, and this made it necessary for biochemists to develop classification systems that help organize and interpret them. Early lipid classification were based mainly on chemical composition and hydrolytic behavior. While these systems were useful at the time, they became inadequate as new lipid molecules were discovered. Historically, lipids were grouped into four major classes based on their composition and the product formed after hydrolysis. They are simple lipids, which are esters of fatty acids with alcohols and yield only these components on hydrolysis. Examples include fats and oils and waxes. Their main functions are energy storage, protection, and waterproofing. Complex or compound lipids contain additional functional groups, such as phosphates or carbohydrate moieties. Examples include phospholipids, glycolipids, and lipoproteins. These lipids are essential for membrane structure, lipid transport, and cell recognition. Derived lipids are substances obtained from the hydrolysis of simple and complex lipids. Examples include acids, steroids, and fat-soluble vitamins. Specialized lipids are involved mainly in regulatory and signaling functions. Examples include prostaglandin, leukotrienes, and steroid hormones. Although this historical system is helpful for introductory learning, it has important limitations. The categories often overlap, lacking chemical precision, and cannot adequately accommodate the large number of newly discovered lipid species. Most importantly, it is not suitable for modern lipidomics and system biology. Advances in mass spectrometry, lipidomics, and molecular biology revealed thousands of distinct lipid molecules. This created the need for classification system that provides standardized nomenclature, chemical accuracy, and hierarchical organization. To address this, the International Lipid Classification and Nomenclature Committee, under the Lipid Maps Consortium, developed a system based on chemically functional backbones. The lipid map system recognizes 8 major lipid categories, which are 1. Fatty acyls, 2. Glycerolipids, 3. Glycerophospholipids, 4. Sphingolipids, 5. Sterol lipids, 6. Prenol lipids, 7. Saccharolipids, 8. Polyketides. Each category is further subdivided into subclasses, allowing detailed structural and functional description. The significance of lipid maps classification lies in the fact that it reflects true chemical diversity, supports lipidomics research, and enhances communication across disciplines. We begin a detailed study of lipids with fatty acyls, which form the foundation of lipid chemistry. In lipid map classification, fatty acyls represent the simplest and most fundamental lipid category. Most complex lipids are either directly derived from fatty acyls or contain fatty acyl chains as part of their structure. Chemically, fatty acyls are derived from fatty acids and their related structures, including compounds formed through chain modification, oxidation, or conjugation. Their long hydrocarbon chains give them a hydrophobic character, while functional groups at one end introduce chemical reactivity. Biologically, fatty acyls serve as energy-rich molecules, structural components of complex lipids, and precursors of important signaling molecules. Understanding fatty acids is therefore essential for understanding lipid structure, metabolism, and function in living systems. Fatty acids are the most important and abundant subclass of fatty acids. Structurally, a fatty acid consists of a long hydrocarbon chain with a terminal carboxyl group. They may be saturated, containing no double bonds, or unsaturated, containing one or more double bonds. The chain length and degree of unsaturation strongly influences the physical and biological properties of fatty acids. Saturated fatty acids are generally solid at room temperature, while unsaturated fatty acids tend to be liquid. Naturally, uh, unsaturated fatty acids usually contain cis double bonds, which introduce bends into the chain. Biologically, fatty acids serve as major energy sources, being oxidized to generate ATP. They are also key structural components of triacyl glycerols and membrane lipids. Certain fatty acids are essential, meaning they must be obtained from the diet, and they play important roles in growth, membrane integrity, and regulation of metabolism. Eicosanoids are biologically active lipid mediators derived from 20-carbon polyunsaturated fatty acids, mainly arachidonic acid. Unlike storage lipids, eicosanoids are produced in very small amounts and act locally at their site of synthesis. Structurally, eicosanoids include prostaglandins, thromboxanes, and leukotrienes, which are formed through enzymatic oxidation of fatty acids. They have very short half-lives and are rapidly inactivated after exerting their effects. Biologically, eicosanoids play central roles in inflammation, pain, fever, blood clotting, immune response, and smooth muscle contraction. Because of their potent effects, they are important targets of many drugs, including anti-inflammatory agents. Their study highlights how fatty acids can function as powerful signaling molecules rather than energy stores. In addition to fatty acids and eicosanoids, fatty acids also include fatty alcohols, fatty aldehydes, and fatty acid conjugates. Fatty alcohols are long-chain hydrocarbons containing a terminal hydroxyl group. They are important components of waxes, and contributes to waterproofing and protective function in plants and animals. Fatty aldehydes contain a terminal aldehyde group and occur as intermediates in lipid metabolism. They are also structural components of certain complex lipids, particularly in specialized tissues. Fatty acid conjugates are fatty acids linked to other molecules such as amino acids, carbohydrates, or coenzyme A. These conjugates play key roles in metabolism, transport, and regulation of lipid utilization within the cell. Together, these subclasses demonstrate the chemical versatility and biological importance of fatty acyls, reinforcing their role as the fundamental building blocks from which lipid diversity arises. Glycerolipids are lipids built on a glycerol backbone, esterified to one, two, or three fatty acids. The most important member of this class are triacyl glycerols, which contain three fatty acyl chains attached to the glycerol. Structurally, the absence of charge groups make glycerolipids highly hydrophobic, allowing them to be stored efficiently without attracting water. They are compact and chemically stable, and they are also energy dense. Biologically, glycerolipids function primarily as long-term energy storage molecules. They are stored in adipose tissues in animals and in oil bodies in plants. Beyond energy and storage, they also provide thermal insulation and mechanical protection. When energy demand increases, glycerolipids are hydrolyzed to release fatty acids for metabolism. The next class is glycerophospholipids. Glycerophospholipids share a glycerol backbone with glycerolipids, but differ in having two fatty acids and a phosphate containing polar head group. This dual nature gives them an amphipathic character, with both hydrophobic and hydrophilic regions. Common subclasses include phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. They are all distinguished by their head groups. These head groups influences membrane charge, curvature, and protein interactions. Biologically, glycerophospholipids are the principal structural components of biological membranes. They form lipid bilayers that define cellular boundaries and organelles. In addition, some glycerophospholipids participate in cell signaling, acting as precursors of second messengers. Their structure is perfectly suited to their role as the architectural framework of cells. Sphingolipids. Sphingolipids are structurally distinct because they are built on a sphingosine backbone rather than glycerol. When fatty acid is attached to a sphingosine, the core molecule ceramide is formed, which serves as the parent compound for most sphingolipids. Sphingolipids can be further classified into sphingomyelins and glycosphingolipids. These lipids are especially abundant in nerve tissues, particularly in the myelin sheath surrounding neurons. Functionally, sphingolipids contribute to membrane stability, insulation of nerve fibers, and cell recognition. Some also act as signaling molecules influencing cell growth and programmed cell death. Their prominence in neural tissues explain their descriptions as neural specialists. Sterolipids are characterized by a rigid four-ring fused structure, which distinguishes them clearly from other lipid classes. The most familiar sterol is cholesterol, which is widely distributed in animal cell membranes. Structurally, sterols are relatively nonpolar but contain a small polar hydroxyl group. This allows them to insert into membranes and interact with phospholipids. Sterols play key regulatory roles. Cholesterol, for instance, modulates membrane fluidity and permeability, ensuring membrane stability across temperature changes. Sterols are also precursors of steroid hormones, bile acids, and vitamin D, linking them to metabolism, digestion, and endocrine regulation. Phenolipids are derived from isoprene units, giving rise to a wide variety of linear or cyclic structures. Their biosynthesis follows conserved pathways that assemble isoprene building blocks into increasingly complex molecules. This class includes terpenes, carotenoids, ubiquinone, and dolichols. Structural diversity is a hallmark of prenolipids, allowing them to participate in many biochemical processes. Prenolipids are highly versatile biologically. They function in electron transport, antioxidant defense, protein modification, and photosynthesis. Some act as pigments, others as cofactors, and others as membrane-associated carriers. The ability to be chemically modified makes them true transformers in cellular metabolism. Having examined the major structural and metabolic lipid classes, we now turn to Saccharolipids and Polyketides, two lipid categories that are less abundant but biologically very significant. These lipids are described as specialized because their primary roles are not bulk energy storage or membrane architecture, but highly specific biological functions. Saccharolipids link fatty acid directly to carbohydrate backbones, while polyketides are synthesized through enzyme-driven condensation reactions that generate structurally complex and often bioactive molecules. Both classes are especially important in microbial physiology, host pathogen interaction, and medicine, providing key examples of how lipid chemistry supports biological specialization. Saccharolipids are lipids with fatty acyl chains linked to sugar backbone rather than to glycerol or sphingosine. This structural arrangement is most commonly found in bacteria and plants, where it plays a crucial role in membrane organization. The major subclasses include lipopolysaccharides and other glycosylated lipid structures. In these molecules, the carbohydrate portion provides polarity, while the fatty side chains anchor the molecule within their membranes. Biologically, Saccharolipids are essential for bacterial outer membrane integrity and barrier function. Lipid A, for example, forms the hydrophilic anchor of lipopolysaccharides and is a key determinant of immune recognition and inflammatory responses in animals. Saccharolipids illustrate how lipids contribute to structural stability, host defenses, and microbial survival. Polyketides are a large and diverse class of lipids formed by the stepwise condensation of small carbon units catalyzed by polyketide synthase. Their structure often contains multiple rings, functional groups, and stereochemical complexity. Major subclasses include macrolides, polyenes, aromatic polyketides, and hybrid polyketide peptide compounds. These molecules are predominantly produced by microorganisms such as bacteria and fungi, as well as some plants. Functionally, polyketides are best known for their potent biological activities. Many serve as antibiotics, antifungal agents, immunosuppressants, and anti-cancer compounds. Rather than serving structural roles, polyketides act as chemical mediators and defense molecules, making them extremely important in pharmacology and biotechnology. Together, Saccharolipids and polyketides demonstrate that lipids are not only structural and metabolic molecules, but also powerful bioactive agents with profound implications for physiology, disease, and drug discovery. Chemical reactions of lipids. Lipids undergo characteristic chemical reactions that are central to their digestion, metabolism, physiological functions, and industrial applications. Understanding this reaction helps us to connect lipid chemistry to biological outcomes. Esterification is the reaction between fatty acids and alcohols, such as glycerol, leading to the formation of esters like triacyl glycerol and phospholipids. Biologically, esterification is essential for lipid synthesis and storage, allowing fatty acids to be packaged in stable, non-toxic form for energy reserve and membrane assembly. Hydrolysis is the cleavage of ester bonds by water, often catalyzed by enzymes such as lipases. This reaction is fundamental to lipid digestion and mobilization, releasing free fatty acids and glycerol for absorption and metabolic use. Saponification is alkaline hydrolysis of esterified lipids, producing fatty acid salts, also known as soaps and glycerol. While mainly of industrial relevance, saponification illustrates the chemical basis of ester bond breakdown and is conceptually important for understanding lipid chemistry. Another important reaction is hydrogenation. This involves the addition of hydrogen across double bonds in unsaturated fatty acids. Biologically, saturation affects membrane fluidity and lipid stability. Industrial hydrogenation alters texture and shelf life of fat, although excessive hydrogenation can generate trans fat with adverse health implications. Oxidation is another important reaction of lipids. An oxidation of lipids, especially unsaturated fatty acids, leads to lipid peroxidation. In biological systems, controlled oxidation plays roles in cell signaling, but uncontrolled oxidation damages membranes and contributes to aging, inflammation, and disease. Antioxidants protect lipids from excessive oxidation or oxidative damage. Other reactions of lipids include isomerization of double bonds and cleavage reactions involved in lipid signaling pathways. Collectively, these chemical reactions explain how lipids are synthesized, degraded, modified, and regulated, reinforcing the close relationship between lipid chemistry and biological functions. To understand the biological importance of lipids, we must first appreciate their role in biological membranes. The Fluid Mosaic Model describes the cell membrane as a dynamic structure composed primarily of lipids and proteins. In this model, Glycerophospholipids and Sphingolipids form a bilayer which the hydrophobic fatty acyl chains face inward, while the hydrophilic heads face the aqueous environment. This arrangement is a direct consequence of lipid amphipaticity. Cholesterol is interspersed within the bilayer and plays a regulatory role by modulating membrane fluidity and stability. The term fluid emphasizes that lipids and proteins can move laterally within the membrane, allowing flexibility and adaptability. The term mosaic reflects the patchwork distribution of proteins embedded in or associated within the lipid bilayer. Biologically, this lipid-based membrane structure maintains cellular integrity, controls selective permeability, and provides a platform for membrane proteins involved in transport, signaling, and enzymatic activity. Thus, lipids are not passive components of membranes, but active determinants of membrane structure and function. Beyond structural roles, lipids are central to cellular signaling and regulation. Certain lipids act directly as signaling molecules, while others serve as precursors of bioactive compounds. Examples include eicosanoids, which are derived from polyunsaturated fatty acids and function as local mediators in inflammation, immunity, vascular regulation. Steroid hormones synthesized from cholesterol regulate gene expression and coordinate long-term physiological processes such as growth, reproduction, and metabolism. Some membrane lipids also generate second messengers, allowing enzymatic modification, enabling cells to respond rapidly to external stimuli. In addition, lipid composition of membrane influences the localization and activity of signaling proteins, thereby indirectly regulating cellular responses. Through this mechanism, lipids participate in cell-cell communication, hormonal regulation, stress and immune responses, maintenance of metabolic homeostasis. Together, these roles highlight lipids as integrators of structure, functions, and regulation, reinforcing their central importance in biochemistry and physiology. While lipids are essential for normal cellular function, alterations in lipid metabolism, structure, or regulation can lead to disease. The pathophysiological roles of lipid reflect how tightly lipid homeostasis is linked to health. Abnormal accumulation of glycerolipids, particularly triacylglycerol, contributes to obesity, fatty liver disease, and insulin resistance. Excess circulating lipids can disrupt normal metabolic signaling and impair glucose homeostasis. Disorders of cholesterol and sterol metabolism are central to atherosclerosis and cardiovascular diseases. Elevated low-density lipoprotein cholesterol promotes lipid deposition in blood vessel walls, leading to plaque formation and reduced blood flow. Defects in sphingolipid metabolism are associated with neurodegenerative and lysosomal storage disorders. This highlights the importance of lipids in nervous system integrity. Altered membrane lipid composition can also affect neuronal signaling and survival. Uncontrolled lipid oxidation results in lipid peroxidation, which damages membranes and contributes to inflammation, aging, and cancer development. Similarly, dysregulated production of lipid-derived signaling molecules such as eicosanoids is implicated in chronic inflammatory and immune-mediated diseases. Lipids are directly involved in respiratory distress syndrome. Pulmonary surfactant is rich in glycerophospholipids, particularly dipalmitoyl phosphatidylcholine. In premature infants, inadequate surfactant production leads to increased alveolar surface tension, alveolar collapse, impaired gas exchange, and respiratory failure. This highlights the vital role of lipids in lung function and survival at birth. Overall, the pathophysiological roles of lipids emphasize that both deficiency and excess of specific lipid classes can disrupt normal physiology, showing or demonstrating the importance of balanced lipid metabolism in disease prevention and management. This module has examined the chemistry, structure, classification, and biological rules of lipids, emphasizing how lipid structure determines functions in normal physiology and disease. We have seen that lipids are central to energy storage, membrane architecture, signaling, regulation, and pathophysiology. To deepen your understanding, the following standard biochemistry textbooks are recommended. Students are encouraged to consult these texts for expanded explanation, clinical correlation, and practice questions related to lipid metabolism and function. This brings Module 3 to a close. Please use the comment section to post your questions, observations, or points that require clarification. Thoughtful engagement will help consolidate your understanding of lipid biochemistry. Thank you for your attention.